Good afternoon, everyone. My name is Karen Chen. I'm an assistant professor in the Division of Life Science. Welcome to the IAS Distinguished Lecture. This is also the inaugural or the first ever student-invited speaker for our division seminar. If you checked your email or are you playing with your cell phone right now, you should have just received a Canvas message if you're one of our postgraduate. You got a survey. You can nominate next year's speakers now. So you can fill in their name, where they're working, what topic do they work on, or you can just tell us what topic of research that you would like to hear from. Like this year, we will fly in the speaker from wherever there is, as long as we can afford it and they have time to come give the seminar. And the student will be responsible to be the host, so you get to spend a lot of time to meet the speaker, and it's a really rewarding experience. So again, I highly recommend you to think about who are the people that you would really like to meet. Please do not be afraid to nominate very high-profile people, or if they seem famous, give it an honest try, and I promise I'll help you along the way. So now you're going to witness the fruit of two students who have worked very hard. I would like to acknowledge. So Veronica is up there. She's sitting quietly, pretending she didn't really do the work. So thank you. And the other is Ada. So I'm going to pass the mic to Ada to do the introduction of our speaker for today.
Well, hello everyone, I'm Ada. I'm sure some of you recognize me. I'm from Dr. Ho Yee's Max Lab. So first of all, I want to thank you, Professor Her, Professor Big Tai, and Professor Karen Chen, to give us a chance for Veronica and I to send out some of these invitations to professors from different places that come and give us some phenomenal talk. So today, I would like to, it's from my greatest honor and pleasure to introduce you Professor Barbara Corkey from Boston University. Barbara received her PhD from University of Pennsylvania in the early 80s. She later on moved to Boston University, and she has been there ever since. And so she wears many hats at Boston University. In fact, she is the Zoltan Kohn Professor of Medicine at the school. She is the Director of the Obesity Research Center. She is also a professor in biochemistry, and she is also the vice chair of research for the School of Medicine as well. I have known Barbara for 16 years now, a very long time. And Barbara has been a very influential mentor in my life. She have helped foster my love for research. She's taught me a lot about, research and, diabetes, obesity, in fact. So Barbara is specialized in metabolic research for the last over 4 decades now, in fact. She has won numerous awards for the contribution of her work in the field, including one of from the American Diabetes Association, the Benting Medal for Outstanding Research in Diabetes. So today, we have the honor to hear about her perspective of how food that we consume have changed over the years may contribute to the epidemic of obesity and diabetes. So please welcome Barbara Corkey.
So thank you very, very much. I'm deeply honored to have received this invitation. And I've had a tremendous amount of pleasure in meeting with the students today, and also in meeting with some of the professors. And I've learned a great deal, because I'm a specialist in metabolic regulation and physiology. And I don't know a lot about molecular biology, but I've learned some very interesting new things. So I thank you. I'm very impressed to discover that it is an international feature that students always sit in the back of the room. It's exactly the same in our institution, and I've never quite understood it, but I'm amazed. So I'm very happy to be here today, and I'm going to talk to you about one of my favorite topics, and it's told in the title, or insulin hypersecretion. I'll explain what I mean by that, and the food additives, a cause of obesity and diabetes, because these metabolic diseases have become the most common diseases in the world. And the incidence, despite many years of research, is not diminishing. It's actually getting greater, and it's spreading to parts of the world that have never had a problem before. And immediately following the beginning of obesity, the incidence of diabetes rises. So the problem, let's see, how do I do this? Okay. The problem is that these metabolic diseases have no cure. We've been working on it for many years, and we really don't understand, although there are many people, and you might even meet them in the street, who think they have the answers. But when subjected to proper testing, it doesn't turn out to really fully explain either of these problems. As a result of modern technology, we've been able to change the expression of a variety of proteins and to focus on a number of different organs, many of which are implicated in obesity or diabetes, but none of which have been proven to be sufficient. And so when a field goes on like this for 20 or more years, it's time to stop and think that there has been little benefit for patients and increasing incidence. So whatever we're doing in our research environment is not beginning to solve the problem. And so perhaps we're not thinking about it in the right way. So perhaps I should stand here. So the common thought, at least in the United States, is that insulin resistance is the cause of type 2 diabetes. However, when you look carefully at the characteristics of the population, you find that there are three things that precede insulin diabetes, and insulin resistance is one of them. But obesity and hyperlipidemia is another, and hypersecretion of insulin. So that means secretion of insulin under fasting conditions where there is no need to secrete insulin. And furthermore, there's no evidence that one precedes the other. Not only that, if you manipulate them separately, you can show that each can cause the others. And for that reason, each of these is a basis for a reasonable hypothesis. And that's what I'm going to look at a little bit today and test the hypothesis, or so begin to test it, that hyperinsulinemia, it can actually cause obesity and insulin resistance. And that's the place we need to look to begin to solve these problems. So for a clinical correlate, what do I mean by hyperinsulinemia, for example? This is fasting insulin levels that were measured in a bunch of patients with different phenotype. So this is the fasting insulin level in normal lean people. As you become obese, express impaired glucose tolerant, become slightly diabetic or severely diabetic, what we see is that the fasting insulin increases. Most people talk about the problem of glucose-stimulated insulin secretion as being characteristic of diabetes, and indeed it is. But if you look at the whole picture together, what you can see is that the percentage of maximum secretion is low in a lean individual, and stimulatory glucose causes a 5-fold increase in insulin secretion. If we go to the next group, this is yet a different group of patients, so they don't look exactly the same. Basal insulin secretion is elevated, as I mentioned before, but now you only get a 4-fold stimulation of secretion. And if you go to the next group, the impaired glucose tolerant, basal is up again, and the fold stimulation is even lower. And now finally, when you get to overt type 2 diabetes, a lot has gone on that isn't normal, and now you've lost even a 1-fold stimulation of insulin secretion. So from all of this, it's perfectly reasonable to test the idea that basal hypersecretion is the cause of hyperinsulinemia, which leads to insulin resistance, obesity, and diabetes. So is this a feasible hypothesis? Well, in this study, these people implanted an insulin mini pump. So this little mini pump secreted insulin for 10 days, and then the animals, these are rats, were tested in a glucose tolerance test. And so the first thing you want to notice is that glucose was perfectly normal at the start of this glucose tolerance. Insulin, as you might expect, because there was a mini pump pumping it in, was about double the normal level. And now when we look at glucose tolerance, first of all, the elevated insulin you would have expected might lower glucose, right? But it didn't, had no effect. It was elevated, didn't lower it. Now you look at the glucose tolerance. Instead of being normal, it's actually impaired. So just simply by giving these rats excess insulin, mimicking what happens in humans who are obese and then impaired glucose tolerant, you can see that we caused impaired response to glucose, and many people would call this insulin resistance, and it was. There was insulin resistance, but we know that the cause was insulin itself by implanting it. Okay, now what happens if we go in the other direction? So you would expect that if you inhibit insulin secretion, which we can do with this drug diazoxide, you would elevate glucose, right? Because you need insulin to store glucose. But wrong. It didn't have, whoops, I can't see which one is which here. One of these is, oh, yeah. This is glucose? Yes. It's perfectly normal. You're inhibiting insulin secretion. And here you can see the inhibited secretion. Here you can see the normal increase that occurs when you, in this case, it was an IV glucose tolerance, you inject glucose. So contrary to what you might expect, taking insulin away doesn't impair glucose tolerance, and it doesn't really even affect glucose levels. So all of this is what everyone believes, and it seems not to be entirely true. Now, I said insulin can cause, itself may be able to cause obesity. And in this experiment, these individuals did sort of an opposite experiment. They inhibited insulin secretion in a group of patients that were put on a diet. And so on this diet, they lost weight, which exactly what you expect if you go on a diet. However, if insulin secretion was inhibited, they lost more weight, significantly more weight, than if insulin were allowed to be normal. And remember, these are obese people, so their insulin level was already highly elevated. Now, a third component of this is with the obesity comes elevated lipid. And elevated lipid itself can have some effects and may, in fact, be a reasonable cause. And so in this experiment, there was a glucose clamp, and you can see on the bottom that both of these groups, the one that had lipid and the one that didn't, have the same glucose level. And on the top, you can see that the normal response to this infusion of glucose is that fatty acids drop way down. But in this experiment, they were infused. And the consequence was that in order to accommodate that increase of lipid, which also comes with obesity, more insulin was required. But again, we've seen now several situations where there's a real disconnect between the insulin level and the glucose level. You can have higher or lower and still have normal glucose. And now we've moved from people and animals into dishes, because it's easier to manipulate dishes. And so these are cultured rat islets. And they're grown either with or without added fatty acid, a fairly high amount. And then Carl Arian in my lab did these experiments for his thesis. He did a glucose dose response, and you can see there's a difference in the shape of this glucose dose response, such that the added lipid caused a left shift in the glucose dose response. Now, a left shift is going to make the beta cell think that it should secrete insulin at a glucose level that's normal. And he did many of these experiments, and I'm going to show you a big summary of that work. And one of the consequences of growing cells, any cells really for that matter, in high lipid, is that they accumulate fat. And so these beta cells were full of lipid droplets. And if we measured the lipid content by staining them with Nile red, what we found that was very interesting is that this, on this axis we have the amount of insulin secreted at 4 millimolar glucose. So this is basal glucose that shouldn't be stimulating secretion. And down here is the condition of cells that are grown at normal glucose without fat. And you can see they're not secreting much insulin at 4 glucose, and they don't have much fat. And as we move up here, we're going up to the top, for example, these cells were cultured at high glucose and high fat, so kind of a normal high-fat diet. And these were cultured at high glucose without fat. These were cultured at low glucose but with high fat. And these were cultured, are these cells that were allowed to recover for 48 hours. And what you can see is that there's a beautiful correlation between the lipid content and the insulin secretion. And so this is consistent with that left shift that I showed you, but it implies that there's a role for lipids in signaling that left shift. So this is basal secretion. What about stimulated secretion? And you can see these numbers, 1 and 2 are being normal glucose, and this being 5 being extreme excessive fuel. So this is now at 12 glucose, which is a stimulatory concentration of glucose. And what you see is this curve took a different direction. So as you acquire more lipid in the beta cell, you actually inhibit glucose stimulated, but stimulate basal. And that would explain why the change from like 5- or 6-fold to less than 1-fold could happen over this range of experiments. But another interesting thing happened here, and that is that cells that were secreting high basal were working all the time, 24 hours around the clock, just like humans who have high basal insulin. So all of that production of insulin amounts to a huge amount and puts a stress on the beta cell. And that stress can be affected in these cells at least rather quickly, and show that the insulin content, the pool from which that secretion came, was also sensitive under the same conditions. And so this continuous stimulation decreased insulin. And this happens actually in humans with diabetes, that their insulin capacity, their capacity to secrete insulin goes down. But it's not because they don't have good machinery. They're producing an awful lot of insulin. It's just not under the right conditions. So the interpretation of this is that obesity, diabetes, and fat can all cause hypersecretion. And hypersecretion and all of these conditions can cause insulin resistance. Either can be primary. So why have we focused only on resistance? I showed you that insulin infusion can cause insulin resistance. I showed you that inhibition of insulin secretion improves insulin resistance and increases weight loss, and that until the very end, normoglycemia is maintained. And that's at least partly because it's the job of liver to maintain normal glycemia. So liver is working just fine. So if hyperinsulinemia is the problem, then what might cause insulin secretion in the absence of a stimulatory fuel? So excess fuel will do that, but that kind of begs the question because, why do you have excess fuel? It's a new problem. suddenly, why does this happen? Well, when we think about the things that have changed since this obesity epidemic began, the first thing that we can point to is that our food today has changed dramatically. There's a tremendous amount of processed food that we encounter. There's some greater than 4,000 new agents that have been added to our food supply, and food coloring, preservatives, emulsifiers, flavor enhancers, all sorts of things. And almost none of these have been evaluated in any way for their effect on metabolism. So we've added all these things, perhaps for good reason, but no one has even begun to really look and see whether any of these things might be related to the problems that we're having today. And if you think that this is limited to processed food, you can't think that very long if you look into things. And so, for example, probably because of standardized growth conditions, improvements in fertilizers and insecticides and what have you, and genetic engineering, even our fruits and vegetables have changed. And so the mineral content of all of these things is vastly different than it used to be. Does that have any impact? We don't know. Again, there has been no testing. In addition to that, unintentionally, we're ingesting a great deal of plastic or chemicals that come out of plastic. So most food is now packaged in plastic or coated in plastic, and that includes the takeout trays, it includes the salad dressings, it includes all sorts of things. Again, we know that some of these plasticizers have been banned because they cause problems in infants, for example, using bottles where the plasticizers get into the, leach out into the milk. But they've been replaced by other things that probably are just as bad because they haven't been tested. So this is just an illustration. When I was a child, ice cream had 3 ingredients: cream, eggs, and sugar. That was it. This is a current ice cream product, and you don't have to read all these ingredients. Highlighted in yellow, I'm showing you those things that might actually be considered food. And there are a few other things there that are not considered food. And now highlighted in pink are some of the things that we've looked at in the lab to ask whether or not they have any impact. So now our model gets a little bit more complex. We have a factor X up there. It might be plastic, it might be food additive, it might be, who knows. And this causes, like that excessive fuel exposure, causes beta cell hypersecretion and leads to all of these other phenomena. So let's look at some of these things. And the way we did this was by developing a high throughput screen to ask whether or not anything caused basal insulin secretion. And we came across, well, we had to stop it actually, because there were so many hits. So many things that we tested had this effect. And sort of like with genetic testing, you find so many variations that you can spend the rest of your life looking at the first 3 that you find, and you still have 300 more left to go. So it was a little bit like that. So I'm only going to talk about the first 3. And the first one was monoacyl glycerides. So these are natural products that are produced in the body and at the cell surface by lipoprotein lipase. But in addition to that, they're commonly used and added to food products as emulsifiers and preservatives. And they were definitely in that ice cream product I showed you. And in fact, in the US, it's hard to find a dairy product that doesn't have this. So what happens when we add monoacyl glycerol to beta cells? What you can see is the concentration-dependent increase in basal insulin secretion. So there's no stimulation here. There's no real work that the beta cell is being asked to do, and yet we're seeing very marked increases in basal secretion. We did something similar with artificial sweeteners. And here in the pink, you see basal secretion. This is at 3 millimolar glucose. And under control conditions, and then with addition of sucralose, aspartame, or saccharin, there's enhanced basal secretion. And in fact, the intermediate concentration of glucose, the response to that is impaired by these artificial sweeteners. And saccharin at very high doses actually inhibited glucose-stimulated insulin secretion. Okay, there's a third one. Iron levels have increased because we've been told that fatty meats are bad, so we should have lean meat rather than fatty meat. And lean meat has a lot more iron, so our consumption of iron is increased. And what happens if we increase iron? The light bar is the basal, and you can see it increases. And unlike the other examples that I gave you, iron also increases stimulated insulin secretion. So they're probably acting by somewhat different mechanisms. But nevertheless, to answer the question at this early stage, what agents cause insulin secretion in the absence of stimulatory fuel? 3 very, very different kind of compounds: monoalesyl glycerol, saccharin, and iron. So, of course, the next question is, how do they do it? And we began to investigate this by looking at the normal pathway that's very well established of how glucose stimulates insulin secretion under normal circumstances. And so it causes an increase in metabolism, an increase in respiration, an increase in redox, an increase in calcium, etc. And we knew all these steps, so we began to look at them. And none of them really had any change except for redox. And this is measuring autofluorescence, which is an indicator of the mitochondrial redox state. And here on the right is the response that you get to glucose. It's a very rapid increase in redox. And here on the left is a kind of similar response to monoglyceride. So we started to think about, this was the only one of the many changes that we know normally accompany glucose-induced insulin secretion that also accompanied monoglyceride-induced insulin secretion. And because we were looking in the mitochondria and because we had done the studies on fuel excess, we thought about this. So fuels generate NADH, which enters the electron transport chain to make ATP. So that's the main function of mitochondria. But what happens when you have excess fuel and plenty of ATP? You have pressure on this end and pressure on this end, and what happens is that you get partial flux through the electron transport chain and the production of reactive oxygen species. So we thought, maybe that's what's happening. And so we looked at it, and here we see the response of beta cells to monoalesyl glycerol compared to basal, and you can see there's definitely generation of ROS, and this is the quantitation. Iron is well known to generate ROS, and this is showing that at both basal and stimulated, you're getting an increase in reactive oxygen species generation with iron and saccharin. This is using a dye that's reflecting ROS generation. You can see that addition of saccharin causes a large increase in ROS production. So these 3 very, very different compounds that cause basal hypersecretion as well as glucose stimulate insulin secretion through the changes in redox and changes in ROS. So it's possible that lots of things can change, but that they're not really the cause. or that they could be a consequence. And so we ask the next question, is this ROS essential and sufficient to cause secretion? And the answer is, you can see here, we're showing the effect of monoglyceride on basal insulin secretion in these two graphs. And in the pink, we're showing the effect of a ROS scavenger. Here it's N-acetylcysteine, here it's resveratrol. And both of these were able to reverse the ability of monoglyceride to generate increased secretion at basal levels. And I can say that we've found similar findings with the other compounds that we tested, but it led us to consider the possibility, well, we could just add peroxide to the outside. And then that's a very clean experiment. And does this have any effect? And as you can see here, this is the basal secretion. This is glucose stimulated, a very nice response. This is the effect of increasing amounts of peroxide added to the outside. And we can also use this compound to generate ROS on the inside. And again, you can see there's a very nice increase in basal insulin secretion. So to answer the question, is ROS essential and sufficient? Answer is yes, it is. So this got us to thinking about implications for other cells. So changes in ROS and redox, for example, in response to these compounds that we added, they're delivered to the beta cell through the circulation. And they happen in response to excess fuel. So that means the same stimulus that's causing hypersecretion in the beta cell is being seen by all the other cells in the body. So, why just think about beta cells? So I started to think about, when does redox change? Is this a common physiological occurrence? And here's a couple of examples. This is in human fasting, and the beta-hydroxybutyrate acetoacetate ratio is shown here. It's a reflection of mitochondrial redox state, particularly controlled by the liver. And let's say in fasting, there's a dramatic change in redox state. And here's another example. Branched chain amino acids are metabolized also in the liver. And if you look at hepatocytes and ask what changes the redox state, well, fatty acids do it. Branched chain amino acids do it. Lactate does it. And the combination of lactate and fatty acid does it even more. So there are a lot of conditions. I'm not going to show you all of them, but included among them are starvation, lean versus obese models on people. So there tends to be in a high-fat diet, an increased redox state. There's some elegant work done by Dean Jones looking at the blood thiol redox state, and he's shown that it becomes more oxidized in diabetes, in aging, in cancer, and in fact, this thiol redox state changes throughout the adult life, becoming more oxidized as we get older. Responses to fuels, in addition to fatty acid, and we've done a bunch of studies doing glucose tolerance tests in people, or insulin clamps in people, and changing their metabolic state also is accompanied by changes in redox. So these are fairly common changes, and they happen rapidly and frequently. So do they change ROS levels? And the answer is, as I've suggested here, that they probably do. Whenever there's this sort of an excess, one sees an increase in ROS, and sometimes one sees an increase in indicators of ROS in the circulation. And just want to show you a real old study. This is from 1973, and this is measuring peroxide in mitochondria in response to all these different things. And let's just focus here on octanoate and oleate, which are fatty acids. And you can see that there's very clearly an increased production of ROS in response to that. So we did another series of studies, perhaps along similar lines, where we had isolated cells in a dish, and we changed the redox state over a physiological range. And we did it with a whole variety of things, but here I'm just showing the cysteine-cysteine ratio and the glutathione oxidized to reduced ratio. So going from left to right, we're going more oxidized. And what you can see is that ROS production, these now liver cells, we're no longer in the beta cell, increase as you go more oxidized. And this change, this redox imposition, is on the outside of the cell. We can do the same thing with other couples, beta-hydroxybutyrate, acetoacetate, and we can do the same thing and have done it in adipocytes, in beta cells, and this is liver cells. I'm sure we could use any kind of a cell and see very similar phenomena. And what this implies is that the external circulating redox state is going to affect all cells, and that fuels or compounds from the environment that change the redox state are going to be communicated to all cells. So in the liver, all right, we see these nice changes in redox and ROS. What about function? Is there any effect on function? And just as one illustration, here we see hepatic glucose production from alanine. And this is with the cysteine-cysteine ratio over the same range that I just showed you, has increased ROS production. And what you can see is that there's very clearly an inhibition of glucose production. So this ROS I've shown you is increasing basal secretion and decreasing glucose production. And in a sense, it's sort of logical. If this is an indicator of fuel excess, then what do you want to have happen? If there's fuel excess in the body, you don't want the liver producing glucose. You want it to stop. But you do want the beta cell to secrete insulin because its job is to store fuel. So both of those things are what I've shown so far today happen. And I can tell you that in fat cells, what it does is stimulate lipid turnover. And it stimulates lipid synthesis more than it stimulates lipid breakdown. And again, this is a logical response to fuel excess. So in addition to the hormonal changes that will be seen as a result of circulation, at each of these individual cell levels, we can regulate very important processes. And I'm not a molecular biologist. You've probably figured that out by now. But this kind of a change is going to lead to major transcriptional regulation. And so for people who understand those things, one can look at the transcriptome, and no one has ever done this as far as I know, over this kind of range of redox, which is physiologically relevant, and expect that we'll see changes in transcription that will eventually result in changes in protein levels and changes in the way the cell functions. And because we're thinking like that, we'd like to propose that redox is a master regulator of metabolism, and that metabolism is an important first step in altering transcription and in altering the protein expression of cells. And so this is a coordinated response to an alteration in metabolic state or a perceived alteration. And it's a very important component of how things are regulated. So for example, I've shown you that liver glucose production can be regulated by a change in redox. I've shown you that beta cell insulin secretion can be regulated. I haven't shown you that fat cell handling of fat can be regulated, but hopefully it'll get published very soon. I've only focused on these cells, but it should be true of all the cells in the body. So there's this communication system that I think we need to take into account when we're thinking about disease states. And I just want to show a few pieces of data from the literature, or information from the literature, making this point. So there's redox sensitive cysteines that are important in FOXO activity. That is an important regulator of many things. There's redox signaling and histone acetylation in pancreas cells. And again, it's not the details of this that I want you to focus on, but just the fact that when redox changes, it can change the activity of all the proteins, of which there are thousands, with redox-sensitive cysteines. So if you want to ask me what's the target of this change, that's a huge problem to directly answer that question, because there are so many proteins that are sensitive. And this is part of the way the cell can work, by being able to share information among many different systems. And I think this is a particularly interesting article by Daryl Neufer in Trends in Endocrinology and Metabolism. And what he's talking about here is that under the normal conditions of the cell, the cells are highly reduced, and in young individuals, highly reduced. And under these conditions, the cysteine sensitive, the sensitive cysteines and phosphatases are all turned off. And that as you go more oxidized, you inhibit these phosphatases. And the redox level at which this happens is different for different phosphatases. And so in a way, this system is having an influence, how controlling we don't yet know, over the activity of a whole variety of kinases. And this is because cysteines are modified by reactive oxygen, which I think I've implied up to now. And so I think it's really important to consider when you're changing things and knocking genes in and out. And I think it's important to consider the metabolic consequences of that, and the fact that there are these generalized signals that can have many effects that aren't really direct effects of what you've done, but a consequence of a metabolic redox change. And this is sort of expressing in a diagram some of what I've been telling you. So in the bloodstream, there are these indicators of redox. So the lactate to pyruvate ratio, which is normally about 10 in the blood, and it's normally about 10 in the muscle. And muscle primarily controls this lactate pyruvate ratio that one observes in blood. The beta-hydroxybutyrate acetoacetate ratio is mainly controlled by liver mitochondria, and it's normally somewhere around 1 in the blood. And the cysteine to cystine, or glutathione to oxidized to reduced, and I think the cystine is believed to be the most important one in the circulation, is controlled by many cells. And these ratios, coming from wherever they're coming from, are also going to all of the other cells in the body. And a final important piece of information is that these are interchangeable. So this is now the inner membrane of the mitochondria. This is the nicotinamide nucleotide transhydrogenase, and it's a transmembrane protein that interconverts NADH and NADPH. And it is active when there's plenty of NADH and when there's ROS. And it is driven by the hydrogen ion gradient across the mitochondria. It's more alkaline inside and more acidic outside. And this NADPH that's produced when this enzyme is turned on is used by oxidized glutathione to regenerate reduced glutathione, which in turn scavenges peroxide and converts it to water. So you can see that if you mess with any of these components, it's going to have an impact on all of the other ones. And so which particular redox state may be doing a particular thing. For example, there's a lot of glutathiolation that goes on that may be important in regulation, but that's also going to be accompanied by other pathway changes that occur because of the redox change. So the road from hyperinsulinemia to redox regulation in ROS, I think that we still believe, and our evidence is consistent with, the fact that hyperinsulinemia could be the problem, and that changes in our environment may cause an increase in redox, ROS, and basal insulin secretion. And we've also suggested and have a little bit of evidence that these changes that we might see in a beta cell can be transmitted to a lot of other places. Well, what about insulin resistance? I started out by telling you that that's the commonly believed trigger for the development of type 2 diabetes. These studies suggest, or our thinking about these studies suggests, that insulin resistance may be beneficial. It may be an adaptive response to maintain normoglycemia in the presence of high insulin. So what would happen if there was no insulin resistance and insulin levels were elevated? What would happen? You'd be hypoglycemic, and that can be lethal. So that would be a very bad thing. So if hyperinsulinemia is the initiating cause, you need insulin resistance to preserve normal functioning. And in fact, if you improved it, you could cause hypoglycemia and a serious problem. And the therapeutics that are currently used to treat diabetes that stimulate insulin secretion may actually be destroying the beta cell, and they may be harmful. And this could explain why the ways that we treat diabetes today never lead to anything that even remotely resembles a cure. Patients just get worse. Patients that go on sulfonylureas that stimulate insulin secretion rapidly lose their ability to respond to that. And so it should be very important to sort out this difference. So my last bit of stuff I'm going to talk about are possible solutions. So one thing we could do is inhibit insulin secretion. And in the beginning, I showed you that people on a diet actually benefited from having their insulin secretion inhibited. But what I didn't show you is that in very limited trials, for example, in type 1 diabetics that have very little insulin reserve, inhibiting their endogenous insulin secretion actually preserves beta cell function. So this has a beneficial effect. It may also have a very beneficial effect in type 2 diabetes, particularly people who have basal hyperinsulinemia. And I suppose if the basal hyperinsulin were low, you wouldn't want to do that. And we could decrease lipid content. I showed you that there was a correlation there. And we know a few good ways to do that. So we could replace long-chain fatty acids with medium-chain fatty acids. So in a dietary sense, this would be replacing, let's say, butter with coconut oil. Or we could stimulate fatty acid oxidation, and we know how to do that by activating PPAR alpha, the transcription factor that turns on the fat oxidizing equipment. Or we could lower lipids by any mechanism that worked. So I'll just show you a couple of things here. So this is using bezafibrate, which is a PPAR alpha agonist. It also has some effect on PPAR delta and PPAR gamma, but believed to have its primary effect on PPAR alpha. And it stimulates fat oxidation. So in this experiment, this is very much like what I showed you at the beginning. We have, this is stimulated insulin secretion in the control and in cells exposed to high fat. And in these cells exposed to high fat, they have a lot of lipid droplets. So in response to increasing concentrations of bezafibrate for, I believe it was 48 hours, what you can see is the ability of fat to depress glucose-stimulated secretion goes away. So you can fully recover it by getting rid of the lipid in the beta cell and by stimulating the fat-oxidizing apparatus. And it hasn't been really tried very much in humans for this purpose. It's been tried in people who have cardiovascular abnormalities, and particularly hypertriglyceridemia. But I think it would be worth a try in pre-diabetics to see if it could serve to prevent diabetes. And here's another interesting experiment where we substituted medium-chain fatty acids over a 48-hour period for long-chain fatty acids. And you'll remember that the basal insulin increased with exposure to increasing concentrations of fat. And what you see here is that didn't happen with the medium-chain fatty acid. And that's because the medium-chain fatty acid is handled entirely in the mitochondria. It doesn't get activated in the cytosol. It doesn't generate long-chain CoA and a whole variety of lipid signals that are derived from that. And it doesn't cause hyperinsulinemia. It also is very poorly stored. So people or mice that eat a diet where medium-chain fatty acids replace long-chain fatty acids tend to lose body fat. So it has some benefit in that regard. So to summarize what I have been talking about here, hyperinsulinemia can initiate insulin resistance. We also show that environmental agents increase insulin secretion in the absence of a stimulatory fuel. And in our current environment, both of those things are perhaps factors. These agents stimulate secretion via changes in redox and ROS, and we've shown that preventing that ROS change prevents the deleterious effect. And we've also shown that, in one experiment at least, that preventing the lipid accumulation does likewise. Since these agents are transported to cells via the circulation, they're affecting or acting in all organs of the body. So our limited vision in terms of the beta cell is not showing the magnitude of the problem, really. And we think that it's important to consider this multifactorial disease due to this communication system that I've discussed. And I need to acknowledge here my collaborators. These are the top line of the people that I've worked with for many, many years. Collaborators are critically important to your success in science because you can't know everything. And it's a combination of tools and techniques and thinking that lead to solution of major problems. And the actual work has been done by the people that are listed here. The people currently in my lab are listed here. And with that, I'd like to thank you.
Thanks, Barbara. Question about when you said that hyperinsulinemia is causal to the diet, or the insulin resistance is compensatory to hyperinsulinemia. I wonder if you now remove the trigger for hyperinsulinemia, would the insulin resistance kind of be normalized?
So we have a really good experiment in humans called gastric bypass surgery. And this is one of the things that has really convinced me to pursue this area. The first thing that happens of all of these changes that we're talking about in response to bypass surgery is that insulin levels drop like a rock. But at the same time, because these people are hospitalized and their diet is highly controlled, the lipid input is dramatically reduced as well. So their food is changing. Insulin resistance, however, lasts much longer. It takes a while to get to change it. And I think the reason is that in response to, these are rather obese individuals who undergo this surgery, and something like 85% of them are cured of diabetes, at least for 5 years. So it makes a number of points. First of all, the lowering of blood sugar and the apparent cure of diabetes happens within a week. The restoration of insulin sensitivity has been induced over a long time, and there are a lot of proteins involved in insulin signaling pathway that are adaptive to this situation. It takes much longer for them to disappear. And I don't know what the exact time course is. So I think that that also proves that the beta cell is perfectly all right. As long as it's not being asked to produce massive amount of insulin around the clock, it is able to recover well enough to maintain normoglycemia. And people who are on insulin go off of it and go off all medication. And as I say, the 85% of them don't have diabetes anymore. So I think that's a pretty compelling argument. Now, there's a lot of arguments still ongoing about, what the precise mechanism of that cure is. And, there are gastric hormones, there's neural inputs that are changed. And probably they all participate in some way. But at the very least, the insulin levels do drop very quickly.
So first of all, thank you for the very interesting talk. So my question is, what would you say about all this from the point of view of type 1 diabeticians who can, in some sense, consciously control their insulin levels? At least they can affect it more than people that don't have type 1 diabetes.
So that's a whole other story. And I really didn't talk about it today, but the responsiveness of people who are going to get type 1 diabetes to lipids and cytokines or inflammatory signals may be different. And that response leads to an autoimmune response, which actually destroys the beta cells. So the only time you have a lot of remaining beta cell function is usually very early at diagnosis, for example. And that's when this experiment was done with diazoxide to inhibit. So they were inhibiting endogenous insulin release, but they were injecting insulin because the patients obviously wouldn't have type 1 diabetes if they had enough insulin. And they did it for some period of time. And what they found was that in type 1, there's something called a honeymoon period. So they get diagnosed, they have very, very high glucose, no control, they go on insulin, and then they acquire good control for a while, and their need for insulin drops. And the length of time that they don't require much insulin can be prolonged by inhibiting insulin secretion. And one of the thoughts that people have is that it's the hypersecretion of insulin early on that's presenting the antigen to whatever the immune cells are that are destroying. So that the fact that they're secreting so much is actually part of the development of the problem. And that really needs a lot of testing. But that's my thought. They are not the same. I mean, obesity in type 1 diabetics is very unusual. They're normally normal weight, and it certainly isn't, triggered by any environmental agents that we know. There's a viral precedent. And so I think cytokines in the circulation probably play a big role. But it's, it has a lot of differences.
Thank you.
Students are supposed to ask questions unless they know everything. And especially if I wasn't clear on something, which I'm sure is true if you don't know this area. So we sometimes do a trick in our university where no one is allowed to ask a question unless a student has asked one. So I'm going to suggest we can't go and have our party until a student asks me a question. Because why are you here?
Thank you for your great talk. And I don't know much about diabetes, but as far as I know that in the diabetes, much of the syndromes that syndromes are reduced from the high glucose in the blood. So I don't quite understand why you suppose insulin, you focus on the insulin hypersecretion, not the glucose.
Okay, so the point that I was trying to make is that one of the graphs that I showed that were data from Ellie Ferranini showed that normally there's, say, 6 to 10-fold stimulation when you go from fasting glucose levels to a high stimulated glucose level. And I was looking at the fasting glucose levels. So under fasting conditions, even in the diabetic and even with the elevated glucose, it's still high. So it's not really the insulin isn't doing its job, that's for sure, and that's why we focused on insulin resistance. But what most of the field has focused on is the fact that the fold stimulation, this 10-fold stimulation, doesn't happen. So when you increase glucose, nothing happens. When it gets bad enough, you just don't get a response. And so, and I think that reflects, and I didn't go into this at all, a failure of liver glucose management. Because one of the things that we didn't talk about is that the liver is producing glucose under conditions where glucose is high. So this is not appropriate. So the liver is not responding, and liver is sensitive to glucose. So an elevation in glucose should turn off gluconeogenesis, and it doesn't do that. So it's not just the beta cell and the insulin secretion that's the problem, it's also the liver cell that's producing too much glucose. It's also the fat cell that's producing too much fat, because that fat is stimulating insulin secretion, and it's also stimulating gluconeogenesis. So now here we have 3, organs that are not operating properly. And the issue, if you want to solve a problem, it helps a lot to know what happened first. So if the liver failed and suddenly started secreting glucose, that would explain diabetes. And maybe the beta cell would try to compensate. But so my suggestion is it's a hypothesis that needs testing, and it's not the only one. I think I could make such as equally good story for elevated lipid being the cause of all of the problems. So the fat cell is the problem. But that's not where I chose to go. And it's been a very fruitful investigation, because if I think that hyperinsulinemia is the cause and I inhibit it, I can prevent diabetes. If I think that insulin resistance is the cause, and there are 2 drugs on the market that supposedly reverse insulin resistance somewhat. Thiazolidine diones, which are PPAR gamma agonists that increase adipogenesis, and metformin, which nobody knows how it works, and every camp has a different opinion. But it definitely improves insulin sensitivity. Well, both of those things also change lipid disposition. So it's not that you're improving insulin sensitivity. What you're doing is you're getting rid of the fat. And in our hands, metformin also scavenges ROS when it's high. So I don't know yet whether that's part of its physiological effectiveness. So if you want to cure something, you have to know what's causing it. And if you focus on the wrong cause, you may get some effects. But it used to be, the reason we have sulfonylureas that stimulate insulin secretion is because everyone believed the problem was the beta cell wasn't secreting enough insulin. So now we make it secrete more. So what do? We kill the beta cell. So this is not a good solution. That may not have been the problem, and that's my, and it's not that I'm, had a better idea than somebody else. It's that they've been testing that idea for 20 years, and it's not going anywhere. And so I think, the important thing to think about is there's always a time to question whether your model is good. You have to always question yourself. And you don't believe, when you begin to be a student, you read a paper and you think everything in there is true. And hopefully, during the course of graduate studies, you learn to be very critical and maybe don't believe much of what's in some papers. And I irritate people a little bit by saying that I think all of the metabolic papers that have appeared, to my knowledge, in Nature and Science are dead wrong. And I'm not saying that the data is incorrect, because I generally believe everybody's data. But the interpretation of what it means, or the importance of what they think it means, is what I think is wrong. So it's a matter, I think that my experience that I shared with you today is one of challenging the existing paradigm and trying to get at what I think is basic cause as opposed to what is consequence. And I think we always have to ask that. So if something happens, is this a cause or a consequence? And you don't automatically know. You really have to think hard to figure it out. So the beta cell failure, which does indeed occur, may not be because of what people think.
Please join me to thank Barbara for the very inspiration talk and her time.